हाँ नहीं करे Hello, good afternoon, students. Welcome back to Botany second year class. So today we are seeing the other FAQs from the 12th chapter. Okay, so one second. Hello. So we have seen these topics yesterday. That is the biofortification and the SCP culture. Okay, and now we will go with the other FAQs. How to change the slide? It's not coming. Here it comes. This is it's coming down. Over. Next slide. Next slide. Now comes the 12th chapter FAQs. That is the what we have already seen is the second FAQ from the 12th chapter. Okay, and now we will start with the most important question, that is the give a brief account of the BT cotton. So yeah, now everyone can say it very clearly. Okay, so in this BT cotton, so whenever this question comes, what we have to write over here, it's the most important one. So BT cotton is a transgenic plant, that is we changed the genotype of the BT cotton variety only. So it became now the bacterial resistant cotton plant. So BT means bacterial resistant. BT means a bacterial resistant variety. So in this bacterial resistant means it is the cotton variety initially is a very disease. It's not the disease resistant variety, but it is getting the diseases very fast by the bacteria and the viral organisms and the fungal organisms. So we need to protect this plant from the diseases that is which are caused by these microorganisms. So we change the genotype of the plant itself in such a way that we can make the completely disease resistant varieties in the cotton plants, because we won't consume this cotton. If we change the genotype also, no problem for our health system, because industrially only this is very, very useful to make the cloth. That means the cloth which we need to cover our body. So we are not consuming it. So no need of the genotype of the plant itself. But we want the much more profits from the field, its field area, for the farmers as well as commercially for the industries also. So for that sake only we made a variety of the cotton which is having the bacterial resistant variety. So in this bacterial resistant cotton variety, which diseases are caused mostly by this cotton plant by the bacteria, cotton bollworm diseases and corn borer diseases are going to be occurred. Okay, cotton bollworm and the corn borer. And corn borer. So these are the two diseases which are caused by the different bacterial organisms in the cotton plants. So we have made a special variety to control these infections in the plant body itself. So through which bacterial organism they will get the infections, and through which we have made it as the bacterial resistant variety, that is the Bacillus thuringiensis. We have taken out the bacteria called as the Bacillus thuringiensis, and we made the plant which is very much resistant for the bacterial infections itself, which are caused by the different bacteria called as the corn borer infection and the cotton bollworm infections. Okay, so for these infections, which one is most important? That is the gene which is resistant for this and causing the infection. That is also very, very important to us. Once we said the genotype is changed, means we need to say which gene is most important one. So in this BT cotton variety, we have taken out their toxinous gene that is meant for causing the infections. So which is coded with the cry gene. Okay, this toxinous gene, that is the harmful gene, is coded with the word as the CRY, cry gene. We'll call it as a cry gene. And here, like this, 3 cry genes are present in the Bacillus thuringiensis. Those are the cry 1AB, cry 1AC, cry 2AB. Okay, cry 1AB, cry 1AC, and cry 2AB. Like this, 3 different types of genes are present. These 3 genes are responsible for controlling the different diseases that are caused by different bacterial organisms in the plant body. So cry 1AB, which can control the disease called as the corn borer infection. Okay, cry 1AB alone can control corn borer infection, and cry 1AC and cry 2AB can control cotton bollworm infections that are caused by the bacteria in the cotton plant. Like this, the toxinous gene, that is the causing the diseases, otherwise controlling the diseases, that is called as the cry gene. And in this, C will be the capital letter. Whenever we are writing the cry gene, the C should be capital letter, RY should be the small letters. Okay, case sensitive this one. And here, how many genes are there? Cry genes, 3 genes are there. Cry 1AB, cry 1AC, cry 2AB. Cry 1AB can control corn borer infection. Cry 1AC and cry 2AB can control cotton bollworm infections. Okay, now this, how to make this complete action of this toxinous gene in the plant body itself. So we have inserted this gene in the cotton plant. So how it protects itself? So now whenever this is caused by the one insect, but that is the insect, otherwise a bird or anything, which comes and takes the cell sap of the cotton plant. Cell sap means the liquid that is present in the cells, that is the cytoplasm of the cell. Whenever it comes and takes the food from the cell sap from the plant body, along with the cell sap, this toxinous genes will go into the insect body, insect mouth. Whenever they goes into the insect mouth is having the alkalinity more. That is the basic nature is more in the insect neck, throat. Okay. So with that infection, that is with that alkalinity more, this cry gene will produces some proteins which will start solidifying over there, makes the crystal-like occurrence. These crystals are giving the rough texture. Okay, rough texture on the in the throat wall itself, inside the throat wall. So with that, the throat will be swollen up for the insect, and which makes it the respiratory problem, and the insect dies. Like that, we can control the infections that are going to be transmitted through the insect to the healthy plant. Okay, so these are the how we have inserted. So same thing, if you explain with the small points, that's sufficient for getting the 4 out of 4 for this answer. So I have given you, I have briefed the complete concept. So what we have to write now, I am saying, okay, because it is the useful for the supply students. So for them, how to prepare it in a proper way, I am saying. So here, once the cryogens and the how many cryogens are there, and how they will control, which type of infection they will control, we have mentioned in the first 2-3 points. Now comes the next point. If you remember the action of the toxin side heading, well and good. If not, also continue with the 4th point. No need of any such side headings for us. Okay, those who are going for the supply exams only, I am saying, because no improvement in the second year right now, we cannot go. So only supply exams where we have to get good score, and we should not lose a single half mark also, not 1 mark, half mark also. Like that you have to prepare. So the 4th point in this is the Bacillus thuringiensis, that is the Bacillus thuringiensis. We can write it as the BT. Okay. If you don't know the spelling of the thuringiensis, if you are writing the wrong spelling, again marks will be deducted. So don't go with the complications over there. Don't make mess for yourself. So here we can write it as the BT. Okay, the strains, that is the gene which is carrying the BT, which is having the cry genes in them. They will start protecting the plant body with a different infectious agents. Okay, that is the lepidopterans, that is the budworm, armyworm. What you can remember, you can say these cry genes, the cry genes which are present in the BT variety of the bacteria can kill the worms, beetles. This one you can write now, beetles you can remember, and the mosquitoes you can remember. You can remember the flies. Simply go with those words. Don't go with the scientific names, otherwise the classes, otherwise the families. Nothing else I need over there. I need only the words which are coming to the concept wise. Okay, cry genes can produce the proteins to kill the different disease causing agents as such as beetles, flies, mosquitoes, etc. No need of writing any other thing over here. Now comes with the how it acts. The BT cotton, the BT gene is having some proteins in them. These proteins will be liquid form in the host plant. Once it goes into the guts of the insect, guts means neck portion, the starting of the neck area. From the mouth, the neck starting part is called as the gut. Once it goes into the gut of the insect, it starts solidifying with the alkalinity of the gut area. Okay, that is the solubilize, means it solidifies into the crystals. Okay, it starts making itself as a crystals occurrence. So once these crystals are formed in the inner layer of the gut area, then it stops, it makes the gut neck to be swollen up. Swollen, we can say now. Okay, swollen up, and it makes the insect to take the oxygen not in a proper way. That means the insect with this swollen neck, insect faces in the breathing, some problem in the breathing, and which leads to the death of the insect. This much you can write over here. Okay, this is the BT cotton. Okay, so in this helps out in the TI plasmid of the Agrobacterium tumefaciens. Okay, we can take out this one, TI plasmid DNA, TI plasmid of the Agrobacterium tumefaciens, which will be incorporated. That means we can insert into the plant body, so that it can even this cryogens will be present in the plant tissues, exhibits the toxinous proteins in the entire plant body, which leads to the killing of the insects. That is because as the cotton is not an edible plant, we can go with this type of transgenic plants, which are industrially very, very helpful for the human beings. Like this, we can conclude. Okay, that means nearly 8 points we have to write over here. So how to give this answer? What describe about the BT cotton? So here BT cotton is the a transgenic plant. We cannot, if you cannot remember the transgenic plant, we can say BT cotton is a plant where we have changed the genotype of the plant body. Okay, by inserting disease resistant gene in it. Okay, and this BT cotton means bacterial resistant cotton variety. Okay, and this bacterial resistant cotton variety can control the different infections that are caused by the different organisms. And the gene, what it will be having in the BT cotton is the crygene, which can control the different infections that are caused by the different organisms. Crygenes are of 3 different kinds. That is a Cry1AB, Cry1AC, Cry2AB. Cry1AB, which controls the corn borer infection. Cry1AC and Cry2AB can control cotton bollworm infections. Okay, these infections are caused to the plant body with the beetles or with the flies and the mosquitoes. And this crygene will be releasing the enzymes in the liquid form in the plant body. Okay, and once the insect comes and takes the cell sap of the BT cotton plant, then this liquid will goes into the guts of the insect. By the alkalinity of the gut area, all this liquid toxinous material will start solidifying, making the crystals on the inner layer of the gut, so that the entire neck will be swollen and the insect cannot take the oxygen. That means the breathing cannot be done properly, which leads to the death of the organism. Okay, like that we can save the cotton plants with the infection that are caused by these insects, otherwise the any other organism. That much we can say. And the ending part, we can say that this cotton plant is not edible, so we can take it out as the transgenic plant because of the very much economic importance is there for this plant. Wind up the answer. That's it. 4 out of 4 you will get damn sure. And the next one is give a brief account of the pest resistant plants. So in this pest resistant plants, again, what we have to write over here in this plants, these pest resistant means these are occurring the several nematodes, that is the parasites. Okay, the nematodes which causes the infections for the different, for the tobacco plants. Through them we need to give the protection for the tobacco plant, that is the pest resistant plants. Okay, the nematodes which gives the infection to the plant body, and those plants need to be protected, that is called as the pest resistant plants. Okay, so here the tobacco plants gets the infection by which nematode? Melodigine incognita. Melodigine incognita. This is the insect name. Okay, melodigine. Melodigine. MELOI is silent. MELOI. Okay, DEGYNE. Melodigine incognita. INCOG. Incognita. NITA. In this, if you are writing the spelling mistakes, 1 mark we can deduct. That much important is the organism name only here. So I am saying how to remember the spelling also, because here we are going with the supply exams. We cannot take any chance right now, na? So I'm saying the spelling also. Melodigine incognita. This is a nematode which gives the infection for the tobacco plant and leads to the death of the tobacco plant, and which causes the great reduction in the yield. Sometimes which leads to the death of the plant also. Okay, which plant is going to be affected with this? Tobacco plants. Okay, Melodigine incognita infects the tobacco plant and reduces its yield and leads to the death of the plant. So how to protect this tobacco plant? Again, industrially tobacco plant is very, very important for us, because in the for making the cigarettes and the and all other things, we need this tobacco plant leaves only. So it's a very, very important plant industrially. So what is the genetic material in this plant? What we read in the other chapters. Okay, that is in the bacteria. What we have seen the SQs from the bacteria. Okay, there we have seen the and the viral organisms, the SQs we have seen. Na? There we have seen the TMV is causing infections for the tobacco plant, which is having the RNA as its genetic material. I said, na, same thing here also we have to remember. And this is having the RNA as the genetic material. Tobacco plant is having the genetic material as RNA. RNA is a single-stranded RNA. Remember, this is a single-stranded RNA. Single-stranded RNA can be seen in the tobacco plants. Okay, so here it is having the RNA, single-stranded RNA as the genetic material. So now it will be the organism through which we are protecting this should have the RNA again. Okay, through that only we can easily protect the plant. And this organism is also having the RNA as the genetic material. So here what we have seen, what we have did, we have adopted it to prevent the infection. Okay, based on the process of RNA interphase. We have taken RNA interphase gene. That is the RNAi gene. RNAi, that is RNA interphase gene. So what happens with this RNA interphase? What is this? It can make a, if the RNA is of like this, a single strand, again, it can make one more strand as the double strand. This is RNA gene and this is RNA. So it makes the double-stranded RNA. What it will do? It makes the RNA as the double-stranded RNA. How it will make? See, so this RNA gene, that is the RNA interface, will be occur. It can do this method in the eukaryotic organisms only. It can be doing this process. So this is the, it involves the silencing of the specific mRNA. Okay. So it silences, that means it makes the mRNA not to go with the translation process. Silencing means it suppresses, it makes it as inactive for a period of time. That is called as the silencing. So we will be having in the bikes, silencers are there now. So what these silencers will do? Silencing mRNA. Silencers, if they are not there in the bike, which is giving the very much horrible noise. If the silencers are there, then the bike, whenever it moves without any sound, isn't it? Just like now the electric vehicles, electric scooters, bikes, cars, which are not at all giving the sound. We don't know that whether the vehicle is coming adjacent to us. When it crosses our body, then only we'll be knowing that yes, that vehicle moved, isn't it? Have you observed this? So whenever we have to pick up is also very good for the electric vehicles. Same thing. Here, silencing. You don't write all those things. Silencing means it stops the action of the mRNA for a period of time. That is the, here the same word in a different way we are using. Silencing mRNA means it makes inactive for a period of time for the mRNA, which stops the translation process. Stops the translation, which is very important over here. Okay, once the proteins are there, means the gene will do the transcription and the translation, and it causes the enzymes, otherwise the diseases. So here we are stopping this. Okay, so with this RNA gene, what it will do? It makes the mRNA to be silencing. Okay, it silences the mRNA. So that occurs by what way? By the complementary double-stranded RNA. What I said already. This RNA makes the RNA into the double-stranded RNA. So with this, what it is doing? It is silencing the activity of the specific mRNA for a period of time. So that binds to and prevents the translation of the mRNA. So this only I said in a simple words for you. Okay, simple words. And how to write also I'll say again. After this, what we have to write? So here the source of this complementary mRNA, that is the RNA gene. Complementary means one more strand of the RNA. Complementary RNA could be from an infection by the viruses having the RNA as the genome. So here this pest resistant plants, which what we are making are the viral infection resistant. Okay, so here this genome should be mobile to the genetic elements and that replicate the RNA intermediate. Through the RNA intermediate, this work can be done. That is the RNA gene will do. That is the second strand of the RNA, complementary to the complementary strand of the RNA will make the mobile genetic elements that stops the, that will be going with the controlling the disease causing RNA genome. Okay, that is the RNA intermediate, through the RNA intermediate. So here, which type of organisms we are using as a vectors? Agrobacterium. Agrobacterium we are using as a vector to insert this RNAi gene into the organism. Okay, into the tobacco plant. So the vector is the agrobacterium, acts as a vector. Okay, and this is the very specific for the nematodes which are causing the infection. Okay, and which will be introduced into the host plant. Okay, so this agrobacterium, through the agrobacterium, we can insert the gene which are present, which are going to be making the complementary RNA strand into the host plant. Okay, and this introduction of the RNA was such that it produces the both sense and the antisense RNA genes. That means it is making the that is the complementary strand. One more strand of the DNA RNA can be made in the host cells, so that now the RNA is a double-stranded RNA. Now the RNA is a double-stranded RNA, which will be initiating the RNAi process, and that silencing the mRNA of the nematode. That is a disease-causing organism. Okay, so with this, we can control the diseases in the tobacco plant, and we can increase the yield of the plant body also. So what we write over here, what we have to write. So you may get a much more confusion by seeing this. Now I am saying what you have to write. So once the question is give a brief account of the pest-resistant plants, immediately what we have to write, the pest-resistant plants are the ones which are causing the plants which are causing the infections with the nematodes need to be controlled, this nematode action on the plant body. That is the pest-resistant plant. Example, the nematodes which will causes the much more damage is to the tobacco plant. So here the nematode is the Melodigine incognita, which causes infection to the tobacco plant and which leads to the reducing the yield in the tobacco plant and leads to the death of the plant body. So to control this, we are taking the help of the RNAi gene. So here the vector used is to insert this RNAi gene. We are taking the help of the agrobacterium as a vector. Through this agrobacterium, we can insert the RNAi gene into the host plant, which will make the complementary RNA strand, which makes the complementary RNA strand. Complementary RNA strand, okay, which silences the mRNA that is present in the nematode and stops the translation process of the nematode gene. So with that, the infections cannot spread in the host plant, that is in the tobacco plant. Like that, we can save the plant life as well as we can increase the yield of the plant also. So this is the one what we have to write over here. Okay. That's the one what we need. But here, what is the most important one? Melodigine incognita spelling and the RNA interference, that is the word spelling is most important. If you don't know this interference, then you can write it as RNAi also. This is also worthful and equivalent to RNA interference word. Okay, if you don't know that, then you can write this RNAi. I is a small letter again, because we are making them as the specific gene which is making a complementary strand over here. That is one strand of the RNA, and the second one will be the complementary to it. So this complementary was made by this RNA I gene. So what we are saying is this only RNA I gene will make the another strand. If you don't know the complementary word, then we can use the word as the another strand of the RNA, so that it will stop the translation process of the infectious agent RNA. Okay, so that is silencing the mRNA of the nematode, so that the yield can be increased and otherwise maintained, and the resistance capacity can be increased for the host plant. And after this, what we have to write at the last, just like the BT cotton. Okay, this one, once we have changed the genotype, this plant is also not edible by everyone. Those who consume cigarettes is the different one. Okay, and the other people, those who are not consuming, we are not come under that category now. So here we should say that it's not consumable by the each and every person in the world. So the persons who are consuming it, they will be having some problems. Health issues will come now or afterwards. Okay, but health issues are coming to them damn sure. If they start consuming this tobacco, they will have this health issues, because in the tobacco plant, RNA I gene is present, which is not accepting by our genotype. Our genotype is a DNA. So that is making the double-stranded RNA, which is not going to be fit in our body. Okay, so here we have to say that this caption at the last, this plant is not consumed by all the persons. Okay, few persons only. Few persons only will do the consumption, not by all persons. So we can easily take this, make this plant, tobacco plant, as the transgenic plant with the first resistant variety, to increase the yield as well as to control the infections in the plant body. Okay, now comes the next one. That is the, now we have finished the 13th and the 12th chapter SAQs. Okay, now we will go with the another chapter. Wait, in this, first question is also there, ma. List out the beneficial aspects of transgenic plants. This we can expect in this supply exam paper. Okay, because BT cotton they have already given. They may give that question again. Otherwise, the pest resistant, or else we can expect this beneficial aspects of transgenic plants. What are the beneficial aspects of transgenic plants? Okay, beneficial aspects means what we are getting the uses by this transgenic plants. So here we have to give this one in the 5 different side headings. Okay, transgenic plants which are made in the crop varieties. That's the one what we have to write. That means once we are growing the plants to get the food for us, those are the crops. Okay, so the transgenic crop plants are having resistance to the pathogens and the pests. Okay, otherwise we can give the heading as the pest resistant and the pathogen resistant plants in the crops. That also we can say. So in this, the examples we have to write. Okay, papaya, you can remember, transgenic papaya is resistant to papaya ring spot virus. Okay, because papaya plant is having the less resistance capacity, but the fruit is very delicious and having the much more vitamins and everything. So here we have to make this as in the cosmetics also we are using the same thing we are saying. So why we are using it will be having some medicinal values. So the transgenic papaya will be the resistant to the papaya ring spot virus. Papaya ring spot virus. And the next one is the what we have already learned, BT cotton. BT cotton is resistant to the insects. Okay, insects resistant. And the next one, tomato, you can easily remember, transgenic tomato plants are resistant to the bacterial pathogens. Example, there is a Pseudomonas. If you remember the Pseudomonas, you can write, if not also acceptable. Okay, so transgenic tomato is resistant to the bacterial pathogens. That much is also well sufficient for this concepts. And the transgenic potato, these are resistant to the fungal phytophthora. If you won't remember the fungal name, no, don't worry. Just write transgenic potato is resistant to the type of the fungi. That's also sufficient. So that you can score at least 3 to 3.5 marks without those examples also. If those examples were written, then full marks you can get. If not, 3 to 3.5 marks you will get. Don't bother for the half mark, because those who are having the problem in going with the spellings, for them only I am saying. Okay, so these 4 points, in these 4 points, if you write 3 points are also sufficient for the first concept, that is the transgenic plants which are resistant to the pathogens and the pests. The first one is the transgenic tomato, which can be easily remembered. Transgenic tomato is resistant to the pathogens, bacterial pathogens, and the transgenic potato is resistant to the fungal organisms, and the transgenic papaya is resistant to papaya ring spot virus. Papaya ring spot virus, like that you can remember. Okay, and the next heading, these transgenic plants are suitable for the food processing technology. Food processing technology. That means where the food is going to be prepared and industrially, where we will use these transgenic plants. In that, the transgenic tomato variety, okay, which is called as the flavor saver variety. Flavor saver variety of transgenic tomato. Okay, this is the one. This transgenic variety will make this tomatoes to be resistant to the infections and increasing the shelf life, as well as delays the ripening in the plant, in the tomatoes itself. So what they are having, they can easily delay the ripening and increases the shelf life. Increases the shelf life. That is the transgenic tomato. That is the flavor saver variety, which is very much important for the industries. That is for delaying the ripening, as well as increasing the shelf life, which is useful in transporting from one place to another place. That is the flavor saver variety. And the third one is it improves the nutritional value in the plant body. So in which way we can say the nutritional value was increased? Here, the golden rice, transgenic golden rice, that is obtained from the type E variety, which is very rich in type E variety, which is very rich in vitamin A. Yesterday also I said, in the rice, if you increases the vitamins, it will be very useful for each and every person, that is a common man. So the same thing here we are saying. Type P variety, which increases, which is having the vitamin A more, that is the transgenic rice. Transgenic rice variety. Okay, so it is a golden rice, what we can say. Type E, which is rich in the vitamin A and prevents the blindness. And the fourth one in this is the with the transgenic plants. What is the fourth one? Which is used in the hybrid seed production. Okay, hybrid seeds are produced for the few plants only now. That is the Brassica napus. So here we have to say it as the plant name is also very important. The male sterile plants of the Brassica napus are produced with this transgenic plants. So what will they will do? They will eliminate, that means they can reduce the manual emasculation process for the hybridization and reduces the cost of the hybrid seed production. Brassica variety is the mustard seeds what we will have, and the mustard oil also we are getting now. So that is the Brassica napus. Okay, so what we are getting with this, we are getting the with the Brassica napus, we are making the plant to be reduce the emasculation, manual emasculation. Okay, reduces the manual emasculation process. Okay, no need for us to remove the stamens from the flower. That process can be reduced by this transgenic plant, that is the Brassica napus. Okay, and after that, what is the most important one? With this, the seed production can be increased and cost of the hybrid seed production can be reduced and the seeds will be more, so that it can be very useful in the outside industries and for the human welfare itself. That is the transgenic plants useful for the hybrid seed production. And the last one is the tolerance to the abiotic stress. Okay, that is the abiotic stress means with the water that is present in the soil. Okay, that is the abiotic agent for the, and the stress resistant means always the water tolerance capacity has to be increased in the plants. So with this transgenic varieties, what we will do will increases the abiotic stress. That is, we are making the plants to withstand the water lacking in the soil, so that they can easily grow if the less amount of the water also, and they can easily resistant to the chemicals and the cold breeze that is present in the outside, in the drought condition, that is the non-availability of the water. Otherwise, the alkalinity and the salinity of the water molecules, everything are the most important one in the abiotic manner. So for all these ones, we are making the plants as the resistant varieties. That is the transgenic plants tolerant to the abiotic stress. In this, the first and the foremost one, what everyone can easily remember, that is the basmati variety of rice. So what will make the biryani? Basmati rice. That is the natural rice that is available in India, and which is a scented rice, which is giving some good smell after cooking also. So plain rice, not the biryani, only plain rice also, it gives some good smell. With that only, we feel much more hungry feeling. Yeah, same thing over here. Now don't go for the lunch. Just have a look of this, then you can go for lunch. Okay. So basmati variety of rice is made for the abiotic and the biotic stress, which is the natural, that is the naturally available plant in India, which is a native of India, we are making them as the abiotic and the biotic stress. That means it can be grown everywhere in India. If the availability of water is present or not in the soil, then also plant can grow, because we know rice variety is needs the 3 months waterlogged field area now. So this variety does not need that waterlogged conditions. It can grow very easily. So it's a profitable for us, and the cost is also good. 1 kg rice is double to the normal rice variety. Okay, not exactly double, but nearly double the price. So we can get much more profit also now. That's the one what our intention originally. Okay, basmati variety of rice made the biotic and the abiotic stress with the transgenic plants. If you remember that, you can write. If you remember the next one also, Roundup ready soybean is herbicide tolerant. Okay, this soybean is the tolerant to the herbicides. That is the one what they are getting the infections. Okay, if you remember this, you can write. Otherwise, only one example is sufficient. Basmati variety of rice. Like this, we have made the other plants are also, which are economically very, very useful. That is the in the petrol industry for making the fuels, and in the pharmaceutical industries, and for making the large amount of the starches from the different plants. We have made the plants as transgenic plants only. Okay, and the next one, these will show these genes will express and they will control the insulin level in the organism body, and the human growth hormones are also somewhat not at all interfered with this transgenic plant genes. So like this, we can make the antibiotics and the antibodies also, and the transgenic plants are very, very helpful for the human welfare. This is the one what we have to write in the beneficial aspects of the transgenic plants. Okay, so with this, we have finished the 12th and the 13th chapter completely for the SAQs. So now tomorrow we'll go with the 14th chapter SAQs and the 9th chapter SAQs. Okay, so till that, stay tuned to this same channel and the same timing. That is 1 to 2 is the class for the seniors in the botany subject. Okay, thank you, students.